Good morning. It's February 25th, and this is your daily brief in science. Here's everything you need to know. A new study shows researchers transferring angular momentum from chiral phonons in quartz to electrons without magnets or external voltages, revealing the orbital Seebeck effect as a new mechanism. The work centers on α-quartz, a chiral crystal whose lattice vibrations carry angular momentum and can mimic a magnetic-like effect. External magnetic fields were used to align the handedness of these chiral phonons, creating a coherent orbital angular momentum flow that drives the orbital Seebeck effect. North Carolina State University led the collaboration, with partners including the University of Utah, and the paper appears in Nature Physics on January 21, 2026, DOI 10038/S41567-025-03134-X. The publication details a multi-institution effort funded by the U.S. Department of Energy, the U.S. Air Force, the National Science Foundation, and international sources. The research advances the understanding of phonon-electron angular momentum transfer and points toward low-power, scalable orbitronic devices for computing and data storage. The approach is expected to extend to other chiral materials such as tellurium, selenium, and certain perovskites, offering lower material usage and longer-lasting orbital angular momentum. α-quartz serves as the test platform, with potential applicability to a broader class of chiral materials beyond quartz. By avoiding magnets and scarce metals, the method points to cheaper, more abundant materials for orbitronics and broader practical impact. Traditional methods relied on magnetic materials and charge injection. The new approach eliminates magnets and external voltages, leveraging intrinsic chiral phonon properties. Direct magnetism measurements in quartz were conducted at the National High Magnetic Field Laboratory, complemented by laser-based optical analysis to observe changes in reflected light. To convert the orbital flow into a measurable signal, the team layered metals like tungsten and titanium on quartz, enabling detection of an orbital Seebeck effect. Aktis Oncology has earned FDA Fast Track designation for AKY-1189, a radioconjugate aimed at adults with locally advanced or metastatic urothelial cancer that progressed after prior therapies, including after Padcev. Fast Track is intended to speed development and FDA review, with potential for more frequent interactions and rolling submissions for a biologics license application. Aktis notes the designation covers patients who progressed after prior treatments and emphasizes the program's focus on an isotope-agnostic radioconjugate platform designed to deliver actinium-225 to Nectin-4-expressing tumors, where urothelial cancer shows high Nectin-4 expression in about 80 to 90% of cases. Aktis is running a multi-site Phase 1b trial of AKY-1189 in urothelial cancer and other Nectin-4-expressing tumors, with Part 1 preliminary data expected in the first quarter of 2027. The trial spans multiple tumor types, including urothelial, breast, non-small cell lung, colorectal, cervical, and head and neck cancers. The company highlights imaging capabilities to verify target engagement before therapy and to confirm treatment delivery, with radioconjugates that can be matched to different isotopes for therapeutic delivery. AKY-1189 is designed to deliver actinium-225 to tumors while aiming for high tumor penetration and rapid clearance from normal tissue. In addition to AKY-1189, Aktis is advancing AKY-2519, a second B7H3 targeting radioconjugate, and maintains ongoing collaboration with Eli Lilly to expand radioconjugate development beyond core programs. Aktis completed an upsized January 2026 IPO, raising about $365 million gross, with equity support from Vita Ventures and a collaboration with Eli Lilly, valued up to $1.2 billion. The press release includes forward-looking statements and warns of risks related to trial timing, regulatory approvals, manufacturing of actinium-225, financial considerations, and intellectual property, advising investors to review SEC filings for formal risk disclosures. A major push to bring quantum entanglement into distributed sensor networks could redefine precision in measurement and navigation. The project aims to push entanglement across networks to boost sensitivity and data acquisition beyond classical limits, with potential uses in atomic clocks, GPS-denied navigation, and magnetic or RF field sensing, and it lays groundwork for a quantum internet. 2 experimental testbeds will validate the approaches: a 25-qubit Rydberg atom array led by Princeton's Jeff Thompson, scalable to hundreds of qubits, and a membrane-based sensor system led by UMIC's Zheshen Zhang, cooled to about 0.1 Kelvin and linked with entangled light. The Rydberg atom platform will use pairs of entangled atoms, while the optomechanical membrane platform will explore continuous variable entangled light fields to connect sensors. The membrane system will start as a single sensor and be upgraded to a 4-sensor network, with entangled light serving as the communication channel between sensors. Researchers from multiple institutions, including the University of Michigan, Princeton, the University of Chicago, the University of Maryland, the University of Arizona, and USC, are collaborating under the Multidisciplinary University Research Initiative focused on distributed entangled quantum sensing. Co-principal investigators span the partner universities, coordinating across disciplines to build the foundation, building blocks, and test beds for discrete and continuous variable sensing networks. If realized, the program could enable quadratic improvements in measurement precision with sensor count, catalyze quantum networking via entanglement distribution, and drive advances in metrology and quantum information science for decades. The goal is to improve sensor network resolution and speed by leveraging entanglement, aiming for improvements that scale with the square of the sensor count rather than the square root. A Nature genetics study shows the early genome architecture is preformed and highly organized before zygotic genome activation, overturning the idea of an unstructured embryonic genome. The genome's spatial organization is a preemptive, disciplined construction that readies transcription at the moment it's needed, not a blank slate. This pre-wiring before ZGA indicates a developmental foresight encoded in 3D folding that supports imminent transcriptional activity. PICO-C's ultra-sensitive method requires only about 1/10 of the input of conventional approaches, enabling detailed chromatin folding studies in early, transient developmental windows and rare cells. A companion Nature cell biology study in human cells shows that removing anchors of the 3D structure can make cells misinterpret structural collapse as a viral attack, triggering innate immunity. The work, conducted in fruit flies, has broad implications for understanding gene regulation, development, and diseases where DNA folding goes awry. The research is funded by the Medical Research Council and the Academy of Medical Sciences, aligning with efforts to link genome structure with regulation and disease. Together, the studies tell a 2-part story: early establishment of genome architecture before activation and health consequences when that architecture collapses in humans. Using PICO-C, researchers mapped the 3D genome in early Drosophila embryos with unprecedented resolution from minimal samples, revealing a modular architectural program of DNA folding. PICO-C shows DNA folds follow a modular program in early embryos, with 3D loops enabling rapid regulatory inputs during development. Developed by Professor Juanma Vacarizas, PICO-C maps genome structure with 10 times less material than standard methods. The preformed genome scaffold primes regulatory elements and genes for rapid, accurate expression, with modular loops acting as discrete regulatory units to maintain genomic integrity during rapid divisions. A modular, adaptable drug delivery platform from the University of Nottingham uses self-assembling building blocks to form RNA delivery nanoparticles via a reversible host-guest linking system, allowing precise tuning of particle stability and behavior. The platform enables safe, efficient delivery of a wide range of genetic medicines through modular components that self-assemble with RNA to form nanoscale delivery particles, with strong potential for rapid development of RNA-based therapies. The work is published in Advanced Materials, with Professor Cameron Alexander among the team, and a contact is provided for more information. The study highlights the platform's potential to accelerate RNA vaccine development during outbreaks, improve RNA therapies in cancer, and broaden treatment options for various diseases. The project is a collaborative effort among the University of Nottingham, Imperial College London, King's College London, University College London, AC. Ltd, and Centillion, Limited. Automated production methods meet stringent quality attributes for RNA vaccines and therapeutics, signaling potential for industrial scalability and rapid deployment. These automated methods demonstrate industrial scalability and readiness for quick deployment of RNA-based medicines. The study appears in Advanced Materials. Copias et al. Modular supramolecular polycations enable efficient delivery of diverse RNA therapeutics and vaccines, 2026, E13315. Experimental results show RNA-loaded nanoparticles deliver RNA across a broad range of cell types with efficacy comparable to or better than leading transfection reagents, and without acute cytotoxic effects. Preclinical tests indicate efficient RNA delivery to diverse cell types, matching or surpassing leading reagents and showing no acute cellular harm. Notes describe the Newcastle Nottingham team's aim toward automated scalable manufacturing and broad applicability across genetic medicines. In vivo in mice, delivered RNAs reduced cancer-associated gene expression in breast tumor tissue and provided protection against H1N1 influenza, indicating therapeutic and preventive potential. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.